Hello, everyone. Welcome to Steeping Heart Health with Green Tea, Insights from Lab to Lifestyle, the next installment of the Linus Pauling Institute's webinar series. My name is Dr. Alexander Michaels, and I am the Institute's Communications Officer, and I am pleased to welcome you here today. I am the moderator for today's webinar, our third webcast of 2024. If this is the first time you've been with us at one of our webinars, I suggest you check out the entire series on our YouTube channel after we're done, of course, not right now. This year alone, we've had presentations on the Mediterranean diet and our top 10 project. In years past, we've done webinars ranging on various topics: vitamins, minerals, phytochemicals, new approaches to healthy aging, and of course, food. Today, we are continuing on the same theme: compounds in green tea and how they influence heart health, with Dr. Richard Bruno from The Ohio State University as our speaker. We have you here for about an hour. We hope to be done today at about 10:30 Pacific time. We're hoping to wrap things up after our Q&A section. Like other webinars in our series, Dr. Bruno will give a presentation for the first roughly half of the presentation, our time together, and then there'll be time for Q&A. And I will talk more about the Q&A later. But actually, I'll just mention right now, there's a Q&A button right below us. And if you ever want to ask a question of the speaker, please use that button. But we'll talk more about the rules of the Q&A as things go along. So let's go along, get things moving, and bring on our speaker. But first, I want to introduce our director, Dr. Emily Ho. Emily, would you like to turn your camera on?
Trying. Oh, why is it
Oh,
not going on? Wait,
and
there
Emily,
goes. Wait.
oh,
Oh no. Hold on.
she's there. She's there. Promise.
Why is this not going on? Oh,
Okay, there she is.
there
All
we
right.
are. Hi,
Morning.
everyone. So if you haven't seen me before, my name is Emily Ho. I am the director of the Linus Pauling Institute, and I wanted to welcome you to our webinar today. It's really great to see some regulars in the attendee list and some new names as well. But bottom line, I want to thank you all for spending the next hour with us. I'm really pleased to have Dr. Rich Bruno here with us this morning. Dr. Bruno is one of our very accomplished LPI alums. And we also have a little bit of a common background. He and I were both graduate students with Dr. Tammy Bray at The Ohio State University. Rich came with Tammy to Oregon State University. I'll talk a little bit about that. About 20 years ago, right about when I joined the faculty at the LPI and OSU as well. So our common mentor and being fellow Buckeyes, Dr. Bruno and I have a lot of things to bond over. We also have a lot of things in common. We love to travel. We both love food. And we also share a common passion for understanding the impact of various foods and their bioactives on health. So as I'd mentioned, Dr. Bruno is a Buckeye. He got his PhD from The Ohio State University, but he spent the last part of his graduate studies here in Oregon. This is where he connected with one of our LPI faculty, Dr. Marette Traber, who brought Rich in as a postdoc researcher. After his postdoc, Rich left for the University of Connecticut, in 2005, but Rich and Marette have continued a long-standing collaboration over these past decades on several research projects around vitamin E, metabolic syndrome, and they've been great partners. So Rich is both a registered dietitian and a nutrition molecular biochemist, and he's able to merge those two skills and expertise to really unravel the health benefits of bioactive food components, including those from green tea that I'll talk about today. He's published over 140 scientific research articles and book chapters. He's given hundreds of presentations all over the world, and is currently the editor-in-chief of the Journal of Nutrition Research. Just recently, Dr. Bruno became the Carol S. Kennedy Professor of Human Nutrition and was also named the Senior Director of the Second Year Transformational Experience Program, also known as STEP, at The Ohio State University. This high-impact campus initiative supports nearly 3,000 students through enriching an enriching mentoring curriculum that's delivered by over 150 faculty mentors. It sounds like an amazing program, and really helps guide students and prepares them for their academic journeys. So, as I'd mentioned, Dr. Bruno has had a long-standing kind of association with the LPI. He and his graduate students have been frequent contributors to our biannual Diet and Optimal Health Conference here at the Institute, and hopefully we'll see him at our 2025 meeting next year. His recent publications on green tea have really hit the headlines as well. So we're really excited that Dr. Bruno could be here today and give us an update on some of this amazing work. So with that, Rich, do you want to turn your camera on? And bottom line, thank you for joining us and telling us about the benefits of green tea. The floor is yours.
Thanks so much, Emily and Alex. It's always a pleasure to connect with my LPI friends. And I'm particularly excited to be here today to share some information about green tea, except I can't share with you because Alex needs to allow me to share my screen. So if you wouldn't mind,
Oops,
Alex.
sorry, got it.
There we go. So as I was saying, I'm really excited to be here to share some science about green tea. Most people believe green tea to be healthy for you. That has been suggested for centuries. But in all reality, the research surrounding green tea has only taken place in the last 20 years. Most of the historical knowledge about green tea is really through anecdotal evidence and folklore from ancient Chinese times, supporting medicinal benefits for green tea. But it hasn't been until like the last 20 years where formal research has taken place. And in this regard, my research team has been somewhat of a leader in this area where we've been examining various health benefits of green tea, but in particular regard to heart disease and closely related cardiometabolic disorders. And we've been conducting many studies over the years in rodents, particularly those that are obese, but also have done some translational work in humans. And you'll see how the two areas of our research are blended together to support this title about green tea, focusing on insights from lab to lifestyle. So I hope to give you a bit of the scientific background, but also some practical translation that you can incorporate into your daily life. So with that, let me tell you where we're going today. Let's not beat around the tea plant and get right to the point. Your takeaway message for today is, yes, green tea is good for your heart health. And I'm going to tell you a story why that is. And as part of that story, I'm going to tell you how it's good for your heart health, the bioactive components in green tea that support heart health, the specific aspects of heart health that it does support, and help to give you some insights of who's likely to benefit from drinking green tea. And as I alluded to a few moments ago, I'm going to share a data-driven story with you based on scientific evidence of the past decades to answer all these key questions. So many of you probably know that tea has a long history of use over the course of many centuries. The origins of tea are actually credited to China because China is the source of indigenous tea bushes and is credited with establishing the first cultivated tea gardens in the world. The use of tea has evolved substantially over the centuries, particularly with each change in Chinese dynasty. Some of the uses have included early on, beginning with medicinal brews, evolving into tonics, cakes, and then eventually grinding the tea leaf into a powder that more closely resembles modern tea that you and I are more familiar with. Interestingly, despite thousands of years of cultural practice and lots of anecdotal evidence surrounding the medicinal benefits of green tea, there hasn't been a whole lot of formal research into its health properties. And this research has only taken place in the last 20 to 30 years in a formal capacity, where we're now starting to unravel the details by which green tea and its bioactive components support health right at the cellular level to thwart disease risk. Perhaps as a ground rule for today, I know many of you have an interest in tea and you drink tea as well as many of the existing herbal infusions. but today's focus is going to be exclusively on green tea, derived from the tea plant Camellia sinensis, due to its unique profile of bioactive components that can support heart health. This is not to say that other varieties of tea or plant infusions don't have medicinal properties, but in the interest of having a focused seminar with some high quality take home messages, we're going to focus on green tea exclusively. So right out of the gate, let me share with you a little bit of some historical knowledge that really ramped up research in the area of green tea. Some of the early work from roughly 20 years ago, some observational studies were conducted in Japan that provided the foundation indicating that green tea could support heart health. In one really, really large prospective observational study involving over 40,000 healthy Japanese adults who were older between the ages of 40 and 79 and were followed between 7 and 9 years, it was determined from history of their diets relative to various health endpoints that the consumption of green tea at 5 or more cups per day of green tea lowered the risk of cardiovascular related death by 15 to 26%. Not dying early sounds pretty good to me. It also found in this particular study that the greater consumption of green tea was associated with a lower risk of stroke, as well as a substantial lowering of the risk of cerebral infarction. Clearly, these are all vascular problems, and what these studies don't tell us is how green tea improves heart health. It just tells us that it potentially does. This is also true of a separate observational study known as the Yoshimi study, another large-scale study in about 1,300 older Japanese men, found that even higher intakes of green tea per day was associated with lower blood cholesterol levels, lower blood triglycerides, and better liver function tests, suggesting that consumption of green tea could support a cardiovascular role. The key thing to recognize here is these studies were observational studies. These are great studies for establishing a foundation of research investigation, but we also have to recognize that these types of studies have pros and cons that counterbalance each other for establishing the health benefits of green tea, as well as any dietary constituent for that matter. On the pro side, these types of studies are relatively inexpensive to conduct, and they're fairly efficient for examining diet health relationships in large populations, such as what I just shared with you in 40,000 persons from Japan. In addition, the large scale promotes a lot of heterogeneity or variation in the study population. This is important because the more variable your population is, the more generalized the findings can be in translating those messages towards human health. On the con side, however, is observational studies are simply that, they're an observation. They're not a controlled trial that allows us to develop conclusions surrounding efficacy and causality. These observational studies are only good for forming associations between dietary factors and health outcomes. And those associations need controlled research, both in humans as well as in rodents and cells, to understand the efficacy as well as mechanisms of action by which dietary constituents can support health. If you see my picture on the right, observational studies fall into this yellow band of cohort studies. As far as quality of evidence goes, consistent with the principles of evidence-based medicine, the quality of evidence is about halfway up the pyramid, and there's different tiers of quality that allow us to fully translate the benefits for any dietary factor. So each type of study has its pros and cons, and at the end of the day, they complement each other to form the strongest conclusions possible. So you'll see this picture come up on your screen again a few times throughout today. Another key concern about observational studies, especially when you're conducting studies in 40,000 persons, is oftentimes, at least as relates to tea research, is it's very difficult to capture the intake of tea accurately. I don't know about you, but I have a hard time remembering what I had for dinner last night and how much of it. Well, reflecting on tea consumption is even more complicated because everybody brews their tea differently. They may use loose leaf, and that's pretty typical in China. And we don't know how many grams of tea leaves that they use relative to the amount of water that they're brewing the tea in. Oftentimes we don't know the source where the tea leaves were grown and the environmental conditions, as well as the temperature of the water that was used to brew the tea or the duration of steeping. All these factors can influence the composition and bioactive profile of a cup of tea. And then, of course, you all know from your own personal experiences, these factors, particularly temperature and steeping time, can influence the flavor as well as the profile of the health protectants in tea. So we'll explore these questions a little bit. What I want to do is help you understand, if you don't already know, that the tea plant, Millet sinensis, is actually used for many different varieties of tea. Perhaps the three most common varieties of tea are green tea, oolong tea, and if you're from the US, black tea is the most common. All these varieties of tea are derived from the same tea leaves from the same plant. But what differs is how those leaves are processed upon harvest. If you're looking to produce green tea from the tea plant, those tea leaves are immediately pan-fried or steamed, and that heat treatment inactivates an enzyme known as polyphenol oxidase. And its activation is really key for protecting against destruction of the bioactive components found in the tea plant. In contrast, teas like oolong and black tea undergo different degrees of fermentation, either partial or full fermentation, resulting in a different flavor signature because these modes of treatment to the leaves either only partially block or inactivate polyphenol oxidase or don't inactivate it at all. So in black tea, this is a fully fermented tea. It's dark black compared to green because of inactivation or not activation of polyphenol oxidase that drives fermentation processes, resulting in destruction of the bioactive constituents, which also provide the flavor profile and the color of your tea that is brewed. Based on that processing, as I'm alluding to, the bioactive component profile varies greatly between different varieties of tea. If you look at my graph on the left side of your screen, we got 3 different varieties of tea. The bottom black section represents the caffeine content across the 3 varieties. Caffeine content doesn't change regardless of fermentation. But what does change is green tea is especially rich in bioactive components known as catechins. And this is certainly not a chemistry class, but catechins are these funny looking molecules that we call polyphenols. The one that's most abundant in green tea is this guy down here at the bottom with the abbreviation EGCG, which is a heck of a lot easier to say than epigallocatechin gallate, which I probably won't say again throughout the course of this talk. But due to its abundance, EGCG is thought to be the most bioactive component of green tea, but that doesn't preclude these other catechins that are prominent from having bioactivities. In contrast, black tea and oolong tea, because they're partially and fully fermented, their catechin content is greatly diminished, such that their catechin content is only about 1/4 that of the levels of catechins found in green tea. And this is why we're going to focus exclusively on green tea today, because we're going to focus on these catechins as the bioactive constituents of green tea that can potentially support heart health, which is the topic of today. Each of us probably brew our tea a little bit different than the other. And the question I always get is, what's the best way to brew your tea so that you can reap the benefits of the bioactive components, but also not create a tea brew that's not too astringent? Based on some very simple controlled studies where tea was brewed at different temperatures or steeped at different temperatures, you can see that if you steep your tea at 185 degrees Fahrenheit, in other words, you bring your tea kettle to a boil, you turn it off, and then you let it cool for a few moments, letting it get down to this temperature, you can maximize the extraction of the catechins from the tea leaves. And you can see that temperature makes a big difference. I call this the Goldilocks rule because if it's too cold, you don't get many catechins out of the leaf. If it's too hot, you destroy catechins coming out of the tea leaf. But if the temperature is just right, you can potentiate that. Similarly, if you steep your tea leaves for between 3 and 5 minutes, that is associated with the best overall consumer acceptability of the brewed tea. And that's true for acceptability as a broad parameter of acceptability, but also specific criteria of color, taste, and aroma. Trained panelists believe 3 to 5 minutes is most ideal for creating the best tasting product. And fortunately, that coincides with extracting the bioactive components from the tea leaves. What's a really interesting concept that I really love sharing with people is certainly we're going to talk about the health benefits of green tea as it relates to people, real live humans. And in fact, I'm going to tell you a story today that relates to their anti-inflammatory, antioxidant, metabolic effects, and also actions at the level of the gut to improve gut health. These are all key features that are supported by the scientific evidence of the modern era. But what most people don't realize is that the tea plant produces these catechins for some key reasons. Unlike you and I, when we sense danger, we can run away. The tea plant, or plants in general, thank goodness, can't uproot themselves and run away from environmental stressors, microbial attack from bacteria, fungus, and other environmental pathogens, nor can they run away from insects that prey on the tea plant leaf and eat it up and essentially destroy the crop. So the plant actually produces the catechins to protect itself from all these stresses. So for instance, synthesis of catechins increases with higher UV light and temperature. It actually has antibacterial and antimicrobial activities. And it also has insecticide properties to thwart insects from attacking the plant. So you can almost think of the catechins as environmental toxicants that protect the tea plant from various stresses. And it's these same low-grade environmental toxicants that provide these unique bioactivities in humans. And these bioactivities are really important because heart disease and atherosclerosis as a specific form of heart disease is the leading cause of death in the United States. And it has been that way for well over 100 years. Some of the key factors that play into heart disease risk include overweightness, dyslipidemia or imbalance between your healthy lipids and your bad blood lipids, hyperglycemia, prediabetes and overt diabetes, high blood pressure, inflammation, oxidative stress, as well as gut barrier dysfunction. I'm not going to go through the complicated picture on the right, but essentially all these risk factors drive key mechanisms that lead to damage to the blood vessels, ultimately leading to the formation of a plaque in your blood vessels that can lead to a heart attack. So we're going to talk a little bit today about green tea as it relates to each of these risk factors and take it home in terms of its protection against heart disease. Arguably, body weight management is perhaps the most important risk factor for reducing your risk for heart disease. I always get the question, can green tea help me either lose weight or maintain a healthy weight? What I can tell you unequivocally is that if you're a rodent who drinks green tea, eats green tea extracts or purified bioactive catechins, the data strongly suggests that you will be protected against obesity. And the reasons for that is, at least in rodents, the bioactive components in green tea are known to have a role in reducing fat absorption by limiting fat digestion and promoting its excretion in feces. Metabolically, green tea catechins increase rates of fat oxidation and prevent the accumulation of triglyceride in adipose tissue. And it also seems to decrease blood glucose, at least by slowing starch digestion and reducing insulin resistance or improving insulin sensitivity, depending on how your brain works. In humans, the question is a little bit more complicated. There have been no shortage of human trials that have been conducted in a controlled manner to examine the question of green tea on weight loss. And because there's been so many randomized controlled trials conducted in the upper echelon of the evidence-based medicine pyramid here on the right, because there's been no shortage of these randomized controlled trials or RCTs, one of the benefits here is that rather than looking at 1 or 2 randomized controlled trials that may or may not show benefits, we now can take advantage of the strength in numbers of these many randomized controlled trials that have been conducted, collapse them into a fancy statistical test and scientific research study called a meta-analysis. Essentially, a meta-analysis is a study of studies where you blend the sum of the parts of various prior studies so that you can look at the totality of evidence for similarly conducted types of studies to formulate one overall strong conclusion, whether or not a dietary constituent like green tea can have health benefits like promoting weight loss. So with that foundation in mind, a few examples are up on the screen. I don't expect you to read this row by row, but there have been a number of meta-analyses done on different types of randomized controlled trials, ranging from studies in women with PCOS, randomized controlled trials encompassing a more diverse population, ranging with healthy and overweight individuals, as well as those with various underlying conditions. Another meta-analysis was also conducted in women with PCOS, and another meta-analysis of 111 randomized controlled trials, 19 of which involved green tea extracts or its catechins, have also been conducted. And then my favorite is you can do a meta-analysis of meta-analysis and blend those studies together to form an even stronger conclusion. So with all that mumbo jumbo in place, if you look at the right column, you can see that there's an arrow pointing down for the outcome of all of these meta-analyses supporting that green tea can support weight reduction. However, what might not be apparent is while weight loss does seem to occur, the benefit seems to be modest, typically in the range of 2 to 5 pounds for all these trials. And the benefits seem to be limited to those who are overweight and consume at least 800 milligrams of catechins per day for at least 2 months. For your benefit, translation of catechins of 800 milligrams per day extrapolates between 3 and 5 cups of green tea per day. Building off that story, dysfunctional blood lipids also contribute to the decades-long process of plaque formation in blood vessels that ultimately lead to narrowing and overt blockage of those blood vessels, leading to a heart attack. And in particular, high cholesterol, specifically high levels of LDL cholesterol, otherwise known as your bad cholesterol, and low levels of your HDL cholesterol, otherwise known as your good cholesterol, drive this atherosclerotic process leading to a myocardial infarction. Many meta-analyses have been conducted examining green tea on blood lipids. To give you a greater insight of how these meta-analyses are done, take this example on the left side of your screen that looked at the effects of green tea in various randomized controlled trials that were blinded and placebo-controlled. Each row in this schematic is a different study that was conducted. The red line is where a neutral or null effect occurs from a statistical perspective. And for each individual study, the black square represents the beneficial effect on the left side of the red line, or not a beneficial effect on the right side of the line. The green line represents the overall mean response across all these individual randomized controlled trials, pointing to an overall benefit that green tea reduces total cholesterol by 5.5 points. Similarly, that reduction in total cholesterol is accounted for by a near equivalent amount of reduction of LDL cholesterol, or your bad cholesterol that really drives atherosclerosis. So it seems very promising that green tea reduces dyslipidemia of your blood lipids. But what's not clear from these studies is what is the optimal dose of green tea? Hopefully I get to answer that question for you today, but across these 20 different studies in this meta-analysis, the range of green tea catechin consumption was between 145 and 3,000 milligrams per day. Clearly, that's a humongous distribution that doesn't really provide you much insight of how much you need to consume to reap the benefits. Building off that, other meta-analyses have been conducted in a similar vein. Corroborating evidence supports, again, that higher consumption levels of green tea catechins in randomized controlled trials correspond to a reduction in total cholesterol levels, LDL cholesterol levels, and a null effect on blood triglycerides. However, when these investigators mined the data a little bit more deeply to look at perhaps responders and non-responders to green tea with regard to triglycerides, it was found that among the 15 out of 31 total randomized controlled trials that examined green tea on triglycerides, the trials that were at least 3 months in duration were associated with a 9-point reduction in blood triglycerides. And then importantly, green tea had no effect on HDL cholesterol, which is a good thing. You wouldn't want your your HDL cholesterol to be reduced because that's viewed as your healthy cholesterol that helps clear tissue cholesterol out of the body to maintain healthy levels of cholesterol. So blood lipids appear to be alleviated in a favorable direction, but what about other risk factors like hyperglycemia and high blood glucose or high sugar levels? The evidence tells us that hyperglycemia is not only a humongous driver of type 2 diabetes. We have 34 million Americans who have diabetes. We also have another 30% of Americans, at least the ones that we know about, who have pre-diabetes. The concern here is not just about diabetes, but most persons with diabetes don't die of diabetes. They actually die most frequently of heart disease because hyperglycemia drives that very complicated progression of disease process leading to atherosclerosis by damaging the blood vessels, inciting inflammation, and driving oxidation of your LDL cholesterol right in the blood vessel wall, ultimately leading to a plaque that occludes the artery. So understanding how dietary components like green tea can manage blood glucose could be important for managing heart disease risk. Similar to the story I told you about weight loss and blood lipids, in humans, lots of randomized controlled trials have been conducted examining green tea in a placebo-controlled manner on blood glucose management. And across 4 out of 5 meta-analyses that have been conducted in recent years in a variety of different populations, there was a significant benefit of green tea consumption to at least modestly lower blood glucose levels. This is really important because it points to green tea having a role in lowering blood glucose in people like type 2 diabetics who already have elevated blood glucose. But in some of these other populations that were included in these meta-analyses, like normal glycemic persons, people with normal blood glucose levels, green tea also lowered blood glucose in those people, suggesting that it may thwart the progression towards prediabetes and diabetes. Getting into the data a little bit more intricately with nuance, one group of investigators conducted a meta-analysis, but wanted to really understand what are the types of characteristics of people who may stand to benefit the most from green tea consumption. It seems like, and I've highlighted in yellow, all the populations that seem to have the best benefit on glucose reduction if they drink green tea. So for example, those who are overweight or obese experience benefit. Consuming green tea for at least 3 months provides the greatest benefit. A dose, interestingly, less than 1,000 milligrams a day was associated with better glucose management, then higher doses above 1,000 milligrams a day of catechins. Seems like both genders are protected against elevations in blood glucose. Perhaps there's a little bit more weight towards females benefiting more greatly. You don't have to be a diabetic in this particular meta-analysis to benefit from green tea, nor do you have to meet the criteria for pre-diabetes, which is 100 milligrams per deciliter of blood glucose, to reap the benefits of green tea. Similar patterns were also observed for the biomarker hemoglobin A1C, which many of you might be aware is an important blood measure taken usually every 3 months because it represents long-term management of blood glucose compared to fasting blood glucose levels on the left. So all this is well and good, supporting that green tea can benefit actual living, breathing humans. but I haven't told you any information about how green tea has these remarkable benefits that support heart health. And the only way I can tell you about that is by telling you a little bit about how green tea influences some of our cellular machinery at the cellular and tissue level. That said, I fully recognize, if I put my registered dietitian hat on, that mice and rodents in general are not men or humans, if I'm being politically correct. But these types of studies provide important insight into the underlying mechanisms by which green tea and its catechins function. I've been trying to do these studies for years in actual human volunteers, but I haven't been fortunate enough to recruit any humans who are willing to donate a portion of their liver or their blood vessel or even their heart or brain for these types of highly invasive studies. But fortunately, the mice show up to work every day, and they're willing to do their part in support of science. And some of the things that the mice have helped us to discover is that green tea catechins can inhibit the activity of digestive enzymes like lipase and amylase that are key for digesting fats and carbohydrates. Catechins promote lipolysis of triglycerides from your fat tissue, allowing them to shuttle their way to the liver and muscle tissue where they can be utilized for energy and burned. Catechins also increase expression of the LDL receptor at the liver, which helps to clear high levels of circulating LDL. Improvements in insulin signaling are apparent at muscle, as well as oxidation of fats through a process known as beta oxidation that generates ATP or energy from fats. Catechins also decrease the synthesis of new fats, a process known as lipogenesis. And in recent years, we're starting to discover that the green tea catechins can shift the gut microbiota in a favorable direction by increasing populations of healthy bacteria while also decreasing populations of pathogenic bacteria. And then when we do some really cool, highly technical studies where we can look at a tissue level, and for example, the liver, we can monitor approximately 5,000 metabolites in response to green tea catechins. And what we can see is something really remarkable that I'm trying to depict on the right side. Rather than looking at 5,000 different metabolites individually, we can pool that metabolic information into a comprehensive analysis where we can cluster the metabolic patterns based on whether or not a mouse is drinking green tea, or I should say eating green tea extract as part of its food, or the combination of green tea extract with a low-fat diet or a high-fat diet that promotes obesity. If you look at the different color circles, you can see that this red box or circle out here represents the rodents fed a high-fat diet. Their metabolic profile is substantially different from rodents fed a low-fat diet, a low-fat diet fed supplemented with green tea extract. When we provide the rodents on a high-fat diet with green tea extract supplementation, you can see that the metabolic profile shifts to something that's somewhat intermediate between low-fat feeding and high-fat feeding, showing that there's broad metabolic effects of green tea on metabolism, which is key for supporting heart health. So building off that story at the liver, my research group has had a long-standing interest in a liver disorder called non-alcoholic fatty liver disease. We now call this disorder MASLD, or metabolic dysfunction associated steatotic liver disease. Just rolls off the tongue. I know you're thinking that. I'll refer to it as NAFLD. But NAFLD is a progressive liver disorder that afflicts roughly 100 million Americans in close association with obesity. And the problem with NAFLD is it results in your liver starting to store fat when the liver is not truly designed to store fat. So the liver becomes fatty, we call that steatosis. Those fatty livers then get inflamed, and we call that non-alcoholic steatohepatitis. This can progress into fibrosis, cirrhosis, potentially liver cancer, as well as death due to liver failure. So this is a really tragic disorder that requires dietary intervention because we don't have any FDA-approved drugs to treat this disorder at this time. So my lab has been very interested in studying the metabolic effects of green tea on liver health in the context of NAFLD. And we focus on these early events more on the prevention side of the more advanced stages of NAFLD. And studying liver health is important for cardiovascular health because persons with fatty liver, unless they progress to liver cancer and liver failure, very few will die from liver-related morbidity, but that doesn't mean it's not a tragic disease. But more importantly, most persons with some form of NAFLD will die of heart disease. So if we can improve the liver, we can improve the heart. Through perhaps one too many studies in mice, we have discovered a lot of profound benefits of green tea over the last 20 years or so. Putting it all together into one slide representing perhaps 20 years of my career, we have found, at least in part, that green tea catechins can help promote a healthy microbiome by shifting the microbiota into healthy populations, as well as suppressing pathogenic populations. In association with that, we see that from the gut to the general circulation, that this bacterial component known as endotoxin, more commonly referred to as lipopolysaccharide in my field, the supplementation with green tea in rodents prevents the absorption of these endotoxins that otherwise go to organs like the liver to activate pro-inflammatory processes that lead to a liver that looks like this. This is a liver from an obese mouse under a microscope. All those white circles are fat accumulation in the liver. And the upper panel is what a normal liver should look like. I would argue you don't need to be much of a scientist to see that obesity has dramatic effects in hurting liver health. But what's really cool is that if you feed these same mice that are obese green tea extract, we can suppress fat accumulation in their liver dramatically. And it seems from our years of research that key to reducing liver fat is some of these interactions at the gut by which green tea catechins inhibit the absorption of these endotoxins by reducing a phenomenon that we colloquially refer to as leaky gut, or the uncontrolled absorption of things through the intestinal barrier. Because between different cells of the intestinal tissue, we have these proteins that connect one cell to the other, and they're called tight junctions, and they do exactly that. They tighten the space between intestinal cells. If these tight junctions are damaged, the space gets bigger, and things can leak directly through the holes in the gut. If these tight junctions are increased in their expression, the gut becomes tightened, and things like endotoxin can't pass through in an uncontrolled manner and accumulate at the liver, where they'll activate inflammation processes that further provoke metabolic disorders. Some of you are probably wondering, well, what if I don't like green tea? Can I reap the benefits from just eating its purified catechins? And the answer is yes, at least in part. We have done those studies in rodents as well, where we fed the rodents a high-fat diet to induce fatty liver. We then supplemented those obese rodents with green tea extract and resolved fatty liver. We then fed purified EGCG at the levels equivalent to that found in green tea extract. And you can see an equaling, if you will, of improvements of liver fat. However, when we feed this other catechin known as catechin, I know that's confusing, but its chemical name is plus catechin to distinguish the two, we see a benefit, but not to the extent apparent to EGCG, at least on liver fat. When we look at other metabolic indicators like body mass and inflammation, we also see a similar pattern where EGCG is, in most cases, almost as potent as intact green tea extract, whereas catechin is less potent than EGCG. So while the catechins appear to be the bioactive constituent, EGCG appears to be the most bioactive, but some of the other studies in my lab also support unique benefits of catechin compared to EGCG. I don't have time to go into that today, but in short, we see that catechin versus EGCG differentially affects the microbiota profile. So there's the possibility of additive and synergistic benefits of a wide array of catechins. So my personal recommendation would be intact green tea or green tea extract, so that you can reap the benefits of the diverse different types of catechins that you can see on the right have not only funny looking shapes, but their shapes differ subtly from one another, which likely gives rise to unique functions. So I'm going to speed up now a little bit and give you some really cool take-home messages as it relates to humans. Because healthy mice don't make healthy humans, we need to find strategies and study the extent to which green tea can improve gut and liver health in people. The challenge here is that most people residing in the United States don't particularly enjoy the flavor of green tea. Most Americans prefer black tea, which is fully fermented and doesn't have catechins present in appreciable quantities. And we know from various studies that at least 5 cups a day of green tea seems to be, quote unquote, the magic level, if you will, of what people should consume to support heart health. It's really hard to get people to drink green tea in general. It's really hard to get them to drink 5 cups of it per day. But what we do know is that Americans want some healthy snacks. So I partnered with somebody here at Ohio State, a food scientist, Dr. Yael Vodevotes, who helped me design a green tea gummy, similar to your gummy vitamins that you might buy at your various grocery stores and health food stores. We developed a low-calorie gummy, provides about 50 calories per day, and at 3 equal servings with breakfast, lunch, and dinner, we can deliver the equivalent of catechins of 5 cups of green tea. We also formulated these confections to be decaffeinated, so people don't have to worry about maybe being overstimulated by caffeine, especially if they're coffee drinkers. And what's really cool about this approach is not only does it have high consumer acceptance from a sensory perspective, but for our randomized controlled trials, it serves as a great way that we can make an equivalent placebo that looks and tastes identically, so we can do studies in a reliable manner. Our first foray into utilizing these gummies was to make sure that people can digest and absorb them and release the catechins, and making sure that they're bioavailable. That part of the question is true. People can indeed absorb catechins from these gummies. But interestingly, when we feed them to groups of healthy weight and obese individuals, you can see here that obese persons in blue absorb catechins over time to a lesser extent than the lean persons. But interestingly, regardless of being healthy weight or obese, the absorption of these compounds known as valerolactones, which are generated by the gut microbiota from catechins, and they are thought to have anti-inflammatory benefit, are absorbed to an equal extent in both populations. So there's a possibility here that catechins and their valerolactones maybe complement each other in their bioactivities to promote various health-promoting bioactivities. The jury's still out on that, and we're still trying to figure that out. So we have used this confection to do some randomized controlled studies in recent years, and one that we just published a few months ago, we used these gummies to examine green tea extract supplementation in people with metabolic syndrome to study gut inflammation, gut permeability, and metabolic benefits. These persons went through a double-blinded, placebo-controlled study where they consumed green tea or placebo for about a month, went away for about a month, and then came back into the alternative treatment, identically as the first time. And what we found is some cool stuff, that green tea extract in the form of these confections or gummies decreased 2 key gut biomarkers of intestinal inflammation in both healthy and obese persons, suggesting that green tea may be reducing inflammation and oxidative stress at the gut that otherwise leads to leaky gut. So then we examined leaky gut in these people by using a really cool non-invasive technique where we can have them ingest these non-digestible sugars and look at the extent to which they're excreted in the urine. And depending on which sugars are excreted, indicates which regions of the intestine are leaky. And what we found was that supplementation with the green tea extract decreased small intestinal leakiness, but not large intestinal leakiness, which is really cool because this coincided with really cool evidence for the first time that green tea supplementation reduced circulating levels in the blood of those inflammation-inducing endotoxins that are implicated in various metabolic disorders like fatty liver. We couldn't get a piece of their liver in this study, maybe the next study, to look at other metabolic benefits, but we did look at their blood glucose levels, and we found that with a month of supplementation, both healthy persons and persons with metabolic syndrome had a reduction in their fasting blood glucose levels. So taken together, I hope I've given you a nice story that green tea is indeed good for your heart health. And this is due to a preponderance of evidence from the bottom to top of the pyramid of evidence-based medicine, with a complement of studies from humans, rodents, and I didn't share you today, but there's a lot of studies in cells supporting this benefit. And now we have some evidence to support the centuries of anecdotes from ancient Chinese history supporting the medicinal benefits of green tea. So next steps and maybe some recommendations for you. We know that green tea reduces various biomarkers implicated in cardiovascular disease. I shared with you very transparently that the reduction was fairly modest for each of those biomarkers, but we've never considered what is the additive benefit of those modest reductions on each biomarker in totally affecting heart disease risk. From observational studies and to some extent in randomized controlled trials, it seems like wide intakes of green tea or wide intakes of purified catechins can have benefits. We don't know exactly what a cup is from those observational studies. And based on my interpretation of the existing scientific literature, it seems that people can reap the benefits with about 3 to 5 cups per day of green tea, which corresponds to brewing green tea with 2 grams of tea leaves, which is what you would find in your grocery store tea bag. My lab is also considering why a range of green tea recommendations may be necessary, because different people have different microbiota profiles that may influence its bioactivity. And perhaps as your last take-home message, because green tea catechins are absorbed and metabolized and eliminated really quickly and absorbed to a very limited extent, you're better off consuming your green tea throughout the day rather than a large dose, perhaps at breakfast, because you need to replenish those catechins that are lost from the body roughly every 2 hours. So with that, I will currently have a duplicated slide. I will pause and stop here for questions. So thank you very much for your attention, and sorry if I went a minute over, but I am here for questions whenever you're ready to shoot them at me.
Yeah, actually, we are a little bit over time. So I think what we're going to have to do is, unfortunately, truncate our Q&A session. But I'd like to thank you for that presentation, Rich. If you'd like to stop your slides. I would love to ask you tons of questions about green tea, but I think I'm going to just limit it to 2 for the time we've got. The first one is about caffeine. Does caffeine make a difference? Do we need to worry about caffeine intake, or is there a benefit of caffeine intake to all of the measures that you've mentioned?
We know from our studies that our benefits of green tea are directly due to the catechins, and there doesn't seem to be any added benefit from caffeine. Okay.
All right. And then the last question is going to be about matcha. What do you feel? What do you think about matcha? I mean, a lot of people love to use this green tea powder. Is it got the same benefit as drinking green tea?
That's a great question. I love that question. So, same tea plant, but before harvest, the tea plant gets covered and it's shielded from UV light. And what that does to make the matcha really, really bright green is it stimulates the production of chlorophyll. So matcha also has catechins, but slightly lower than that than green tea, or I should say tea that has not been shielded from light, because UV light stimulates catechin synthesis. So there's some trade-offs. If you're trying to consume more chlorophyll in your diet, matcha is great. There's also some questions about when you grind up the tea plant for matcha, the extent to which ingesting those particles of tea leaves actually releases the catechins during digestion. That has not been fully resolved yet.
Okay. All right. So as far as we know at the moment, drinking green tea is the better approach.
Yep.
Well, at this point, we're going to have to call it because we've unfortunately run out of time today. Thank you, Dr. Bruno, for joining us today and giving us a new perspective on green tea. I wish I could just run out and get a cup right now and steep it for 3 to 5 minutes.
Thank you, Alex and LPI friends, for having me today. It was a pleasure.
And I'd like to thank everyone in the audience for your great questions today. As always, we've had more questions than we have time for answers. So I suggest that you look for our next LPI Digital Digest, which should be out in the winter, probably in January or February, which will address some of those questions that we didn't have time for today. Any final words for our audience, Emily?
Well, thank you, Dr. Bruno. Thank you, Alex. Thank you, everyone. I appreciate the time you took to listen to our webinar today. And it was just an amazing webinar talking about the benefits of tea, and I learned so much. As Alex mentioned earlier, the programs here at the Institute are completely supported by donations from people like you. I know many in the audience are already donated. So thank you very much. But if you appreciate the webinars and the content from the Institute, please consider giving a gift today. Here's the slide in terms of information to give. And bottom line, if you'd like to get more and more information about the Institute, there's lots of ways to do that. The LPI Digital Digest just was published last week. It includes previous webinar recaps, extra Q&As from our speakers, and a copy of our top 10 brochure. You can find them all of this information on our website, but if you'd like a print copy of any of these materials, just let us know. Lastly, everything related to LPI, again, can be found on our website, lpi.oregonstate.edu, as shown here. It includes the Top 10 project, our newsletters, other timely information. You get links to our webinars there as well. Speaking of webinars, our next webinar should be coming this coming fall. So watch our website and your emails. If you're not on our email list, sign up and you'll get all the information and these announcements as well. So thank you again for spending time with us today. We'll see you next time. Stay healthy.